So thank you very much, everyone, for joining us. This is another Academy of Nutritional Medicine AUNM webinar, and it's about understanding Toxiplex. And we're delighted to have Kunal Garg here with us, together with Professor Leona Gilbert. I'll introduce Kunal. He's a biotechnology and nanoscience expert specializing in diagnostic solutions for infectious and chronic diseases. He's the co-founder, CTO, and shareholder of Texted Limited, a Finland-based in vitro diagnostic manufacturing company. He leads the development of innovative ELISA-based multiplex tests, including those for tick-borne diseases, viral reactivation, autoimmunity, and mycotoxins. And it's the mycotoxins that we're going to hear about tonight. So, Kunal, I'll leave it to you to say anything else about yourself you'd like to.
No, thank you. So I'll start with the presentation. Thank you, Julian.
With the slides, yes. Can I just mention to everyone to put your comments and questions in the Q&A or the chat, and then we'll have lots of time afterwards for answering them. Thank you.
Excellent. Okay, so I'll just share my screen, and please let me know if you can see it. So you can see it?
Yes, that's fine.
All right, great. A very good evening to everyone. Thank you for being here. So we'll be discussing today about Toxiplex, and so I'll be taking you through the modes of exposure and the analytical workflow of Toxiplex, and by the end of the presentation, you will have an understanding of the type of samples that you should be using for measuring mycotoxins and the modes of exposure that you can think about in your clinical practice, for example. So I do have a disclosure statement. Obviously, we are talking about one of the products that we manufacture. So this is essentially my disclosure statement. And what I will be discussing today is I will be going through essentially the One Health concept and then discussing the modes of exposure for mycotoxins. There are many different modes through which we get exposed to mycotoxins. Finally, I will be introducing the concept of human biomonitoring, especially in the context of mycotoxins. And we will talk about specifically urine versus serum sample, the difference between them, and then the different test techniques and what they tell us about the measurement for mycotoxins. So before we talk about mycotoxins, let's just take a step back and talk about fungi. So fungi play an important role in the environmental health, human health, and animal health. And therefore, it is one of the key suspects when it comes to One Health. When we talk about fungi, we don't really associate it with only the negatives, because fungi do help in improving the soil fertility and also pest control. But also, when we talk about the negative sides of it, they do play a role in diseases for plants, humans, animals, and especially what the topic that we will be talking about today is the food spoilage. But since mycotoxins are the secondary metabolites of fungi, we also need to see the positive side about the metabolites, which is them being used in the pharmaceuticals and the biotechnology industry. But obviously, we're here to discuss about mycotoxins and the contamination of mycotoxins, which actually leads to food spoilage. I introduced the concept of One Health because One Health is not just about what is going on today, but it is also about the future. And in the future, there are papers and research to show that climate change is inducing more aggressive species of fungi, which are playing an important role in terms of producing mycotoxins that are more harmful. Now, these don't just affect the plant health or the animal health, but they play an important role in endangering the food security and ultimately the human health. As we were discussing, when we talk about mycotoxins and the different modes in which we get exposed to it, the primary mode is through food, and that is through oral exposure. The food, as we've discussed, gets contaminated because of the air, water, and land that is essentially containing fungi or mycotoxins in some or the other form, which then leads into food. There is good documentation to show that 80% of the crops, either before or after harvestation, contain some or the other form of mycotoxins. Now, the issue with mycotoxins is that they are very hard to process because they are stable at very high temperature, and you can always smell mold, but you cannot smell mycotoxins. Yes, the presence of fungi indicates probable presence of mycotoxins, but mycotoxins are very, very small molecules. We don't essentially know which mold it is and which mycotoxin it is producing. The other method to get exposed to mycotoxins is in the occupational health space, wherein you can get exposed through inhalation. And this is in the agriculture space, warehousing space, and also food processing space. And I will be discussing more about both of these. So mycotoxin exposure through food is very, very well recognized in the US through FDA and in the UK, for example, through Food Standard Agency. And you will see some of the same culprits, for example, aflatoxin, ochratoxin, and then these are the ones that are very important to notice because these are the primary suspects when we discuss about mycotoxin exposure through food. And I will be discussing these later on in my presentation. But when I say uh, mycotoxin uh, exposure through food is very well established, I mean really well established. And as you can see over here, this is a map from 2024, wherein it's an annual report that is published every year, wherein each country submits a certain number of samples and then these samples are tested for different mycotoxins, and the level of each mycotoxin is reported. So I will take you through this so that you can just appreciate the variety of mycotoxins that we see. So in 2024, roughly 28,000 samples were submitted by 95 countries. And you can see that the darker the red color, the higher is the percentage of risk of mycotoxins in that particular country. So the highest risk we see is in China, wherein there is in the submitted samples, 90% of them had mycotoxins, whereas compared to Australia or the Oceania, we had 9% of them with mycotoxins. The other thing that you will see over here is that not a single it, one mycotoxin is not fitting all of them in all the country. For example, in Oceania, fumonisin is the one that is highly prevalent, whereas if you look at Central Asia, deoxynivalenol is the one that is highly prevalent. And when you go on in different continents and in different regions, you will see that the profile of mycotoxin is really changing. But one of the things that we should be really aware about is the co-contamination. And this is something that I want to really press on is that 70% of the samples that were submitted in 2024, 70% of them had more than one mycotoxin. And I just want to press on this because when we think about multiple mycotoxins, for example, in a food product can be infected or contaminated with multiple species of fungi. Each of the species of fungi produces multiple mycotoxins. And if a particular food product is contaminated with multiple fungi, you can only imagine the number of different mycotoxins that a particular food contains. So it is not unprecedented that when you measure mycotoxins in a particular food product, you detect multiple mycotoxins. And therefore, when a particular human consumes such contaminated food, they would be contaminated or they would be exposed to multiple mycotoxins through a single food product. But as we discussed, food is not the only form of exposure. Uh, Household and indoor environments are also a mode of exposure. So this is a study from 2022 in Germany, wherein out of 24 households, different building materials were measured for mycotoxins, were measured for mold. And you can see that certain species of mold really stand out, that are highlighted in red. And each of these species actually produces multiple mycotoxins. So, for example, penicillin produces patulin and ochratoxin, and the Aspergillus species of versicolor, it produces aflatoxin, steric ochratoxin, and gliotoxin. And so it's really a roulette wherein, depending on the mold species, which can be multiple mold species, you get multiple mycotoxins. So it's just increasing in terms of risk. Here are some papers to further make it evident that in wallpapers, in cardboards, in and in other places in indoor environment, for example, in industrial area, if there's a corn dust, then you see different types of mycotoxins represented in each of these indoor environment. So does indoor environment play an important role in mycotoxin exposure? It actually does. So this is a very good paper, and this was from a wet waste management, wherein they had taken samples from the air, from the surfaces, the blood sample of people working in that area, and also there was a personal air sampling for these people, and the sample was also taken for them. Now, there were people that were working in spring, and then there were people working in the fall. And you can see that there was actually no difference between people working in the spring versus fall. But of course, there was a difference between people that were not working in the facility. So when you're working in a facility, or if you're in an environment where there is mycotoxin exposure risk, then you see that in test results also. So what exactly is the risk of mycotoxins? Well, aflatoxin can disrupt the liver, and it is a suspected carcinogen for hepatocellular carcinoma. For DON, it is known to disrupt the gut, and there is also research coming up wherein it affects the brain and gut axis. For fumonisin, we see that it can inhibit the adaptive immune response. So the dendritic cells and the maturation of the dendritic cells are inhibited. For ochratoxin, they play a important role in the nephrotoxicity, and also they cause inflammation. And finally, zearalenone, it can mimic the estrogen molecule, and it can also inhibit the synthesis of proteins. So just a summary of the exposure slide. So we get exposed through various inhalation, skin, or ingestion, and these can come from a variety of sources. Now, when we are getting exposed to these things, for example, DON and FB1, you can see that they can affect the immune system, digestive system, liver, and kidneys. And what is more concerning is that there are certain mycotoxins like aflatoxin and ochratoxin that are present more in baby food and tomato products. And this is important to note because when mycotoxin exposure happens in adults versus in babies or young kids, the amount of exposure becomes highly vulnerable because the exposure is always measured as a total weight. So since the weight in young children is less, the amount of exposure becomes high as percentage of their body weight. So going through the human biomonitoring concept, so when we do get exposed to the mycotoxins, we can either use urine samples or serum plasma samples to measure the mycotoxins. And to measure the mycotoxins, you can either use liquid chromatography mass spectrometer or an ELISA test. Now, the ELISA test, I just want to make it clear that I am only dealing with ELISA tests that are directly detecting mycotoxin molecules and not the IgG, IgE, or any other antibody response towards the mycotoxins. So there is a lot of Since we've started working on Toxiplex, we've received a lot of questions from patients and from doctors regarding uh, urine tests for mycotoxins versus serum tests for mycotoxins, and they are usually compared. And I want to make a point that these tests should not be compared. Rather, these tests actually work in complement, and they are really for different uses. So, in the urine, what you're essentially measuring is that if the body is eliminating the toxins or not. And in serum, if you catch mycotoxins in the serum, then it is essentially a persistent contamination that is affecting the body. So when you talk about exposure window for mycotoxins in urine, it is anywhere between 24 to 48 hours, and it is best for catching acute cases. So it is really is the body detoxifying as it should. But when we talk about long-term cases, for example, ochratoxin has a half-life of 36 days almost. So it is detectable for a long period of time in plasma if it is a persistent mycotoxin that is affecting the body, and it is not something that is so you use serum when you want to measure persistence of mycotoxins, but you use urine to measure if the body is detoxifying or not. And this is also from the levels. So a particular urine test for mycotoxins would be traditionally positive because you're essentially measuring the detoxification, which is measured anywhere between 24 hours to 48 hours. Whereas if you're measuring it in serum and if you see it in serum, then it is usually when the levels of mycotoxins are changing very slowly. Now, the advantages and the limitations in urine versus serum is that urine, the sample collection is very easy. You can really, because it is non-invasive, you just pee in a cup and then you have the urine sample. For serum, yes, it is slightly invasive, but it tells you if there is a chronic effect of mycotoxins or not. So essentially, when to use urine is when you're expecting a recent exposure and if you're expecting the clearance of mycotoxins, if it's happening or not. But if you're expecting an ongoing or chronic exposure, that is when you use serum or plasma. So if a urine test is positive, but a serum test is negative, then it essentially means that the body is detoxifying and there is no ongoing exposure. But if the urine test and serum is positive, then yes, there is some level of detoxification, but there might also be a ongoing exposure. If only serum is positive and urine is negative, that is actually an interesting case, and we've not seen that as of yet. But yeah, it is a possibility. So moving to the type of tests that are used for measuring mycotoxins, I just want to make it clear that we are not dealing with culture-based or molecular-based tests because those are used for detecting fungi or fungi. And for mycotoxins, because these are small molecules or these are small metabolites, you can use chromatography methods. This could be either mass spec or any other readout. And the 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 positive aspect of this is that you can get multiple mycotoxin detection. But the negative part about this is that chromatography machines are not readily available, and it takes really uh, highly trained personnel to actually perform these tests. What we are focusing on is antibody-based tests. And I know when I say antibody-based test, the immediate reaction is, are we measuring IgG, IgM, IgE? No, we are not measuring any of those. So what we have designed is a mycotoxin test wherein we are detecting, like a chromatography test, the mycotoxin molecule directly. And so we are not measuring the immune response to it. We are measuring if it is in serum plasma or not. So most of what we do, we try to publish. And of course, in 2022, we published our multiple mycotoxin detection in human serum. And this is an open source paper if you would like to read it. But I will just go through some of it. So again, just to make clear, we are directly detecting the mycotoxins and we are not detecting the IgM, IgG, or IgE responses. And also, we selected to do it on serum or plasma and not on urine, because in urine you need to standardize it, because the urine volume from patient to patient can be different. Also, we are not trying to measure if the natural detoxification process in a particular specimen is working or not. But we are trying to establish the ongoing exposure in patients. So just to give you an idea of how this test actually works. So when we make the test, we immobilize highly specific antibodies on a particular microplate. And then these plates are sent to, for example, a lab like Almond Labs, wherein they extract the mycotoxins, these black structures, and they compete it with synthetic mycotoxins. Now, if the synthetic mycotoxins are low in quantity, then it means that there is detectable mycotoxins in the serum. But if it is not, then it means that there is not detectable mycotoxins in the serum. So the mycotoxins that we focus on are aflatoxin, DON, FUM, OTA, and ZEA. As you would remember from my previous slides, these are the main suspects usually when we are dealing with mycotoxin detection. And we have 93% accuracy and also very low analytical detection limit. So I just want to go through again a lot of numbers, but I will just go through the comparison of detection limit. And this was actually one of the challenging parts, and this is from the publication, because when we were trying to publish, we did not have a precedence of any other researcher or research group actually developing a multiple mycotoxin detection kit for use in human serum. So we actually had to compare our test to well-established food mycotoxin tests, because they use the same principle. And you can see that in most cases, our detection limits are much lower than tests that use the same principle. Of course, when we are talking about a antibody test, antibodies are inherently prone to cross reaction. And as you can see that when we have an aflatoxin antibody reacting to a DON molecule, FUM molecule, or the other molecules, then we only have 2% of a cross reaction. So the minimum cross reaction that we've seen is 2%, and the maximum that we've seen is 11%. And this, by an immunoassay standard, is expected, but we have acknowledged it in our paper. So between different classes of mycotoxins, we do not see that high of cross reactivity. So in summary, the modes of exposure to mycotoxins is high. We can get exposed through food or through indoor environment. And if you remember, a particular food product or particular indoor condition can have multiple molds that are contaminating it, which essentially means that there are multiple mycotoxins. Therefore, human monitoring of mycotoxins is important. But again, recognizing and appreciating whether using urine sample or serum plasma sample is important. So urine sample would be for acute exposure and the detoxification and to understand the detoxification, wherein if you're trying to understand the chronic exposure, then that is where the serum and plasma comes in. And what we have been able to essentially create is a best of both worlds. So we have created something wherein it offers multiple mycotoxin testing and high accuracy, like the chromatography techniques, but then we can also make it highly accessible and high sensitivity, which is expected from ELISA tests. So that was all the slides from my side. Thank you.
No, thank you very, very much indeed. Very interesting, lots of information there. Do you want to stop sharing and then we can? Thank you. So there are some questions, and a few were asked in advance as well. So I'll start with Suzanne, who asks, is this a good test for ongoing mold exposure in a water damaged building? How do we know whether the exposure is from food or from contaminated air if the environmental exposure is not immediately obvious?
So how do we know whether the exposure is from food contaminated? Right. So at least the mycotoxins that we have included in the tests are prone to both. We see them in food and in indoor environment. So I think it is very difficult to say if it is from food or indoor environment because we are just measuring the mycotoxin, and the exposure of it needs to be established either by the doctor or by checking the mold. Because you can smell mold sometimes. So if it is through mold, then it might be actually something that has been going on for a while, and you already have an idea that it might be in my surrounding. But it's very difficult to say that from the test. We can only measure the mycotoxin and say that if it was detected or not. And yes, the Toxiplex is for ongoing because we have optimized the test for serum and plasma. So yes, it's for ongoing.
Well, thank you. And particularly in the States, traditionally urine mycotoxin tests are used. She's asking, why do so many doctors rely on urine mycotoxin tests to monitor treatment for long-term mold exposure?
Well, we ask the same thing sometimes, to be honest, and that was one of the reason why we actually went in a different direction. Because if you, and I'm getting a little scientific here, but when we look at PubMed and the number of articles that are published for human biomonitoring of mycotoxins, you will see that a lot of researchers are using serum and plasma. There are papers on urine, but it is for acute exposure. So it might be, I mean, from a benefit of doubt standpoint, it might be that the doctor is trying to understand if the patient is having a natural detoxification process or not. But if a patient understands that serum is for long-term exposure and urine is for short-term exposure, maybe the patient can have that discussion with their doctor to have a better understanding of the process. But yes, we do not have the understanding of why urine is used for long-term exposure. Yeah.
Yeah, and maybe I can add to this that, as Kunal mentioned, urine is a non-invasive sampling technique. Okay, so it's simple. It makes it practical for repeated monitoring, but Armin Labs has a solution to this non-invasive sampling, and to make it a very simple by the use of Tasso. So if you have not been to, I think you might actually with AOM, you might actually have this information on Tasso. It is a gadget, a blood draw gadget that you puncture in your arm. It's very easy, it's very simple, and very reproducible with the use of Toxiplex with these samples. So maybe because of legacy testing, doctors are used to, sending urine, getting urine and sending it in. But nowadays, I think it's a lot more sensitive and practical to use Tasso and to use Toxiplex to really look at treatment success. Yeah, and Kunal is showing the Tasso device that Armin Labs is offering for this type of testing.
Yeah, that's very simple to use, isn't it? And I think you've checked the sensitivity and
Yeah.
specificity, and it's almost 100%, isn't it?
Yeah. There's no difference between this and the normal phlebotomy and Tasso.
Yeah. Oh,
Yeah.
thank you. And then I'll just go back to the questions because there were some others.
Mhm.
The same therapist says in her experience, some doctors use elevated markers on urinary mycotoxin tests to justify ongoing antifungals. Is that correct, or is peeing out the mycotoxins actually a sign that you're getting rid of them and that you shouldn't necessarily treat really intensively?
So if you're peeing out mycotoxins, that essentially means the natural detoxification process is working.
Yeah.
Yeah. So I mean, on treatment, I have to say I cannot comment because I'm not an MD. And so the specifics of the case would be different, and I cannot make a general statement on that.
Right. Are there cases where high toxin loads show up even in asymptomatic individuals?
Yes.
Yeah.
Yeah. I mean, asymptomatic individuals would be understandable because firstly, the exposure in itself is complicated. Are you getting exposed through food or through indoor environment or both? And asymptomatic is a possibility because mycotoxin is not a first line test. So patients probably go through a lot of pain before a particular doctor says that, oh, it might be actually mycotoxin. So yes, I think asymptomatic conditions are possible.
And the second part of the question was, if so, how is this best interpreted? I mean, simply by seeing a high load in the test, would you say? It turns red, doesn't it, in your test when there is a load showing?
Yeah. So the symptom, so Toxiplex aids in the detection of mycotoxins. I think how it needs to be interpreted actually depends on the correlating symptoms also. And usually patients in our experience that get tested with mycotoxins also have a bunch of other tests that have been done. So it's actually the interpretation of the test is not just based on Toxiplex results, it's based on probably fungal tests that have also been done or infection-based tests that have also been done. Some patients get tested for Candida and things like that. So it's really a whole package. But yes, if you're seeing mycotoxins in serum plasma, uh, uh, which is quite unique, then there's definitely been an exposure of some kind and it needs to be followed up. Yeah.
Good question.
Gillian, I think you're mute.
Oop. Or do you want me to take over?
I'm sorry. No, well, yes, that would be super if you'd like to after this. But I was reading this question here, which is, what is the recommended frequency of testing with Toxiplex in ongoing chronic illness management? In other words, how often would you recommend doing it to see whether the load is decreasing?
Uh,
Yeah, that's a good question.
Yeah.
Very difficult questions.
Yeah.
Uh, I think I think the well, I mean, in terms of balancing the cost and not doing overdiagnosis, it really depends on how frequently the person sees the person sees their doctor. I think that would be the best way because I'm a test producer, but at the end, we don't want to cause over anxiety in patients. So a moderate frequency would be best discussed with their doctor in this case.
Yeah, depending on what else is
But
going as well. Go ahead. Yeah.
yeah, exactly. Yeah, I was just going to make mention to that. There, because treatment is so personalized, okay, and if the practitioner wants to, let's see, binders, the effects of binders, okay, and getting rid of mycotoxins, and want a follow up every month or every 2 months or every 3 months, okay, then I would have that discussion if I was the patient, saying, okay, then let's discuss what type of test then to really show, if you're going to see me every month, how about, let's say, every 2 months or every 3 months, and make that agreement with the practitioner, based on also symptoms and based on also those other standard tests that are being performed for the patient themselves. Okay, like kidney functions for one, because if the kidneys are not working, obviously, even sampling of urine is going to be difficult, or if the person is immune compromised, or if some other, systemic dysfunction is occurring, then that's going to obviously affect the treatment success. So I know the answer. I don't like answering, well, it depends. It really does on the person and the whole picture of the person. So if we have a case study specifically for a person with symptoms, with all the tests, this test, and then the proposal treatments, and then we can, consider what type of testing regimen should be done and in what frequency.
Thank you. And is Toxiplex complementary to the tests related to detecting the condition known as CIRS, CIRS, or can it be used instead?
I think it's a complementary one, okay? And we are not published with this one, but it is a complementary one. And I think though there's also some other like blood markers that we could biomarkers that we could test for to complement this to really look at the toxics, environment that the person has. So it is, I really believe a complement one, not a replacement, but a complement one. But let's also rely on those standard tests like TGF beta 1 or MMP9 testing, the C4A testing, those standard testing that would come with this type of disease, let's use that and get the whole picture and then this and complement it with if the person really is uh, dealing with the toxic environment that they have.
Right, well, that makes sense. And what are the most common findings you see with Toxiplex? Are there particular mycotoxins that you see in specific countries, for example, where you're testing? And how do they correlate with symptoms? I know that's a tough question, isn't it?
Yeah, there's a lot of interest. That's good. The most common we see is OTA, and we're still compiling a lot of data because we keep getting the data and we keep compiling it. But the most common we see is OTA, and this is also in
The
line.
most common is what? Sorry, I missed that
Sorry,
word.
ochratoxin. Ochratoxin.
Okay.
Yeah. I use the abbreviation, but
Mhm.
it's ochratoxin A, and this is in line with the published research by other researchers, for example, in Spain, wherein they had studied, I think 700 or 800 patients, and they measured them for probably 30 mycotoxins, and the most common one came out to be ochratoxin A. So we are seeing the same pattern. For country specific, we have not been able to do that as of yet because it's going one dimension lower in the data analysis. So we've not gone in that depth at the moment, but maybe in future AONM webinars we can also have that the same map as we have for food, but a little less comprehensive. And for symptom correlation, that's another dimension that we are trying to also get because we are test producers. We do not have access to patient samples directly, which we have, for example, with Almond Labs. So from Almond Labs, we can easily get the test results. But then I say another layer or another dimension because Almond Labs does not get the symptoms directly. The doctor has the symptoms.
Yeah,
So
exactly.
so the traceability of that, it really gets lost, and we end up with a very small sample size for such analysis.
But I do encourage all practitioners using Toxiplex if they really want, really good solid statistical analysis of to answer these questions to please, contact us so that we could, collect the, symptoms, okay, with the test results. Okay, we can do a research finding on this and hopefully get you published and us published and working together to, bring statistical analysis of Toxiplex into the scientific world as well as the clinical world even more because we need that and we can help you publish your data sets and your patient cases on this because we're really interested in it, of course.
Very, very good point. Thank you. I know you provide so much support in putting together studies. Thank you. Another very good question here, though again difficult, is what advice might one give to a patient to minimize their exposure to mold in foods? Is it simply going for organic foods? I know that some therapists, even quite prominent ones, say that organic foods do tend to be more susceptible to molds, or certainly some kinds. What's
Yeah.
your sense there?
Well, I don't know about organic foods, but there are some aspects that do turn out. For example, in the Middle East, there are certain mycotoxins that stand out. For example, aflatoxin, because they are present in nuts a lot, and sometimes also in dates. For example, so there are certain products of food that are inherently always having mycotoxins in them. So, for example, in Europe, example for Finland, I can speak about Finland. There's usually high levels of deoxynivalenol in the oats. So the oats that are imported over here. So if you're in a particular country and these someone like me would go into Finnish customs data to look at it, these data points are not like really out there, but it would be very interesting to just Google uh, what are the most mycotoxins that are seen in UK and in which products, and then try to avoid them as best as possible. But please do note that most food products do have mycotoxins, so that should not lead you to the ultimate conclusion that eating food is going to cause you mycotoxin exposure, because of course there's detoxification process also that our body can handle.
Of course.
And
Yeah.
and I would go a little bit one step more and take Kunal's, global map of which countries have the highest, mycotoxins and correlate that back to which food groups actually have those okay, and then avoid those. Like as Lucille says here, like corn has high mycotoxins in it. It's true. Corn, wheat, oat, barleys, peanuts, pistachios, cashews. So it's not necessarily organics. It's more of what food source and where it's coming from. So that's that's the point, to avoid those types of high mycotoxins in those foods.
Sure. And she also says that surely retesting would ideally only be needed if the diet and environmental reasons were being addressed.
Yeah.
Would you like to go on with a few more questions, Leona, because I've got this
Sure.
off at the moment. Thank you.
No problem.
So
Yeah, no,
thank
I
you.
think that some of these questions are very practical and they're very informative even for me. Like, is there any advice on storage of foods? I mean, the science demonstrates that, of course, dried, airtight environments for most of the grains and the nuts, so that, no moisture can get in. I think that's really the point. Also, they are suggesting it is suggested that also freezing like corn and oats and barley, if you're not using them, okay, immediately. So I think it is important to think of airtight containers, freezing, dry places, that kind of thing, and also uh, uh, moisture absorbers. Okay, if you're long-term storing corn or dried corn or popcorn or barley, I like use moisture absorbers to make sure that there's no possibility that mold can grow
Yeah.
or fungi can grow.
One thing to add over here, uh, it's always best practice that if you've seen fungi in a particular food that has been contaminated, then it needs to just be thrown out, because there
Yeah,
are some people that might think that a particular slice of bread, if it's been spoiled, then and it's not on the others, that is really not the case, because fungi has roots, so the mycelium, and those are invisible. So if it's in a particular package, then it's as good as bad.
So throw in a
yeah.
little of them then if
Yeah,
it's an
yeah,
apple or one strawberry. Yeah,
yeah.
get that. Yeah.
Yeah.
Are you planning to expand the range of mycotoxins you test? I know at one point in a previous webinar you said you were possibly going to do a second panel, or do you think that's not necessary? Are we basically hitting on the key ones here and one shouldn't make things too complex?
So
Yeah.
we always want to expand. There's really no limit to the amount of mycotoxins we can test. I mean, the Toxiplex is a smaller panel compared to Ticplex, what we are doing. So we can test for more mycotoxins, but I think what we need to do is we need to balance between the needed and the cost. Because, for example, if a patient wants to do retesting, we need to make it affordable enough that it is not a burden. And this is something that we get actually from Armin. Armin is like, you cannot overtest, you cannot undertest, you have to just be at that very small optimal space wherein the clinician is happy and the patient is happy. So we're trying to balance that. We're trying to get more data from or feedback from clinicians and data from Armin labs to make that decision. But we do have our eyes on a few mycotoxins to work on. Yeah, we always do have some to work on. Yeah.
And I just want to also point out too, like I know this is kind of like geeking out because we're medical device manufacturers, but there is a limit to actually what we can do because either there's antibodies against the mycotoxins that don't exist
Hmm.
yet, okay? There's not enough research to validate addition of certain mycotoxins in the test kit. Also, there is no legal governance anywhere in the world, in any country in the world, legal governance in testing, diagnosing mycotoxins, in humans, Okay, there isn't. There is for animals and feed, but there isn't for humans. So, this is an emerging area, and we're on top of it, which is great. So uh, we've had frequent conversations with our health authority. Not only in this country but in other countries, because the law is just not catching up to this emerging field, and up until that point, then we have to just kind of be cautious in what we do, meaning and also follow the science and in the limitations of the science, because there is major gaps in this area. And if you talk to legacy clinicians, they find it difficult that mycotoxins could be responsible for certain diseases like immune dysfunction and so forth, or
Absolutely.
kidney liver failure and stuff. So there is an area of large gaps, in mycotoxin diseases. And because of that, there's no laws. Because there's no laws, the science is lacking. And because the science is lacking, we can't, have all the reagents that we need to make the perfect test.
I know for,
We understand what, yeah.
yeah, no, I was just
Sorry,
going to
go
say,
ahead.
I know for you and also for Armin Labs, validation and certification is particularly important, and you wouldn't want to bring out tests that can't be backed up by the science and certified. And I know that Tickplex and Toxiplex aren't they, completely 100%.
Yeah, and for Toxiplex, I mean, because there is no governance, we wanted transparency from our side. So actually, before we launched the test kit, the paper was published, and the multiple mycotoxin in human serum, that's been cited many times, and that was the whole point, because we want, and it's open access, so anyone can go there and actually see what we are using and what the readout is, how it was measured, and the how the limit of detection was calculated and all of that.
Right.
Yeah,
Go
so the
ahead.
verification, the validation is completed on this
Yeah.
test kit. It's just there's no legal governance for use of this, or any mycotoxin test for human diseases. There's not even the ICD codes, those international codes of diseases,
Oh,
for such diagnosis. There isn't. That area is lacking. That's why this is emerging. So, yeah, but we're on top of it, and we're watching, and we're trying to change the landscape of this for
yeah.
the patients so that they can get better diagnosis and better, options for diagnosis and treatments. So
Yes, thank you. There was another question which came in earlier, um, because you mentioned how often patients have done many, many different tests before doing a mycotoxin test. Are there particular types of patient or clinical case where you would recommend Toxiplex as a first line of investigation?
Well, if there is known exposure, definitely that would be the first line. But in our case, and in discussion with the people at Almond Labs, we've heard people getting tested when they are suspected of immune suppression.
Mhm.
So, and a test that is usually ordered also with Toxiplex is the Candida test. So
Oh,
if patients are getting tested for some sort of fungal related infection or opportunistic infections, then they're usually tested for Toxiplex also. So in those cases, and the third is patients that have been tested for mycotoxins in urine. So there are definitely some practitioners that understand the difference between acute and the long term, which is something that we have a discussion with them on this regard. And that is actually where we got this understanding from of why they use urine versus serum. So those are also patients that do get tested to see if the natural detoxification versus persistence of mycotoxins in
that
serum.
makes a lot of sense. Yes, thank you.
and also, if I may add too, that if you have a patient that is experiencing autoimmune flares, or immune flares and immune dysfunction, flares, This is a very good indication that there's something else going on, okay, than other underlying infections, okay, and it could be immune-related mycotoxin disease. And this is the thing I want to point out here, that we should be looking really at the immune fitness of these individuals, okay, and the flares, the symptoms, the flares that come up. And also chronic fatigue syndrome patients, okay, there's a high correlation of mycotoxin exposure to these types of patients as well. So that would be another indication to use this in first line testing.
Thank you. Would you like to just mention any upcoming events that you'll be presenting or speaking at? I know that ILADS, the International Lyme and Associated Diseases Society in Portugal, which is from the 1st to the 3rd of May. I'll be there as well.
Oh, great.
I hope I've got the date right. April, May, yes. So I look forward to seeing you there. But are there any other exciting events coming up that you'd like to mention?
Yes, yes. Well, Kunal, you can talk about your talk in
Yeah, so
ILADS just shortly, and then I can go.
I'm basically presenting Toxiplex, or not Toxiplex, but I'm presenting a mycotoxin test at ILADS, which is going to be the same presentation. So I will be talking about mycotoxins at ILADS, basically. And after that, well, Leona is going to many events.
The one in
Yeah,
Finland
but
is taking place for the first time. Will you be covering mycotoxins there as well?
No,
See,
actually, we are covering big data and AI and crypto infections, chronic pathologies. So not really mycotoxins there.
Not really. Okay.
Yeah.
but let me talk about this event here since you asked. I'm going to share screen if that's okay. Yeah. So we have this Lyme Disease Awareness Month coming up in May, and we have the privilege of actually screening the Red Ring documentary that has been made from this gentleman. He's a patient, Jonas Berghall. He's a film director, writer, and obviously chronic Lyme disease patients. And he made this documentary a few years ago. And we, like I said, we're going to have some screenings of this documentary for free for people, for free. And we have 6 screenings, 2 in Europe, 2 in Australia time zones, and 2 in the Americans time zones. And if you're interested in watching this documentary for free, it is being sponsored by AONM from other partners like Armin's Lab and so forth, and Tandem Clinic, Labomed Issue, and others. But if you want to come here, go to our website here, tested.com, and the Red Ring, and pick the date that you want to register. Okay, and there's also an in attendance raffle that we're going to have free testing offered by Armin's Lab, and it will be Toxiplex Plus testing, and 3 people that are present at the viewing is going to be chosen for this free test, and in each of these viewings. So not only is the documentary really informative, because actually Jonas went to WHO and the United Nations, okay, to discuss about chronic Lyme, but there's familiar faces in this documentary. So we have the privilege to build awareness around chronic Lyme disease through this documentary, and hopefully we'll have you guys attending, and possibly lucky attendees can get some free testing. So this was the happening in May, and also there's one showing in July, but we also have another event. And as Jillian, as you mentioned, we have this conference, the first annual conference of chronic infection pathologies.
yes,
It's really going to be, yeah, it's going to be in Finland. Okay, we're here. And it's the theme is to show me the data. But what's really unique about this conference is we wanted to be united in the sense of patients, practitioners, and also scientists coming together, uniting, and trying to change the landscape of chronic infection pathologies. It's not just chronic tick-borne diseases. There's other pathologies there, like long COVID, chronic fatigue syndrome, etc. But this is the first conference, and will be on the 5th to the 7th of September in Uvascular, where we are right now. and it is quite unique and affordable. There's incentives for patients and students and early career scientists and practitioners and even medical students for the registration costs. Abstracts are open. So if you think that you have some data that you want to show, please submit an abstract. And yeah, we have the event happening on Thursday to Sunday, and we have some networking events as well. But the main point is that we have a cost reduction for patients. We have cost reduction, obviously, for speakers, for students, for early career scientists and practitioners. We want to support them as well to attend for this. And this is the first conference that's really bringing in all three kind of important stakeholders in this area. So I just wanted to mention this kind of conference here. Yeah.
That sounds really, really exciting. Thank you. And we'll put notifications of that in our newsletters and in the e-blasts that we send out. Thank you, Helga. I think the question came in a little bit later, but you've answered that partially for now.
Yeah.
And there's just one aspect of it that I've heard before as well, which is a claim that lime sometimes comes out when polystyrene is used for mold. Is that something that you've heard? Or is that fairly new? She's just
Does
that make sense?
I have not heard.
I have not
seen the data for that, I have to say. But I mean, we're open to seeing such data if it exists.
Yeah. Well, thank you so much. That was a fascinating presentation, Kunal, and thank you so much.
Thank you. Thank you,
Leona, too, for all your contributions. Just to mention that we'll actually be seeing you again on the 14th of May in a round table where you'll be presenting a lot of cases, I believe, related to Lyme and also pandas. So that will be fascinating. And so that will be in one of our newsletters that comes out very, very soon, for e-blasts. And we do have a lot of other events going forward, so please watch out for our information on that you'll be receiving. So any final words, Leona or Kunal, before Easter that you'd like to just
No, thank you for your time and thank you for the opportunity. This was great. And we really like doing AONM webinars. So
thank
we'll
you.
be back.
Is chocolate with Easter coming up? Is chocolate traditionally very tested with mycotoxins?
Oh,
it's the only prediction I have.
It is? Oh, okay. I suppose it depends where it's come from and what kind it is.
Yeah, exactly.
Well, thank you very, very much.
Thank you.
And thank you everyone who's attended today. And there were questions about whether this would be was being recorded and would be put up on our website. Absolutely. And may we have your slides as well, Kunal?
Sure.
Thank you. So the slides will be available too.
Yeah.
So thank you very, very much, everyone. Have a lovely time off over Easter coming up, and see you again after that. Thank you.
Cheers. Thank you, guys.
Bye.